Good afternoon and welcome to our webinar this afternoon. Before we start, just a little bit of housekeeping. If you have any technical issues, please send a short message using the question box on the right hand side of your screen. We'll try and answer the technical issues. Sometimes we're not able because it's to do with IT issues, but we will try. During the webinar, we would like to invite you to ask questions using the questions box, which is also on the right-hand side. And at the end of the webinar, time permitting, we'll try to answer as many of these as we can. If we don't get around to all of the questions, we will answer them offline and we'll share them with you along with a copy of today's slide. So today's webinar is on microbial identification technologies and developing an ID program. So, my name's Annette Russell, and I'm the sterile and non-sterile microbiology lead here at RSSL. I've worked in the commercial team for over 7 years, and my role is to support our clients with a variety of microbiological testing, from routine microbial limits through to more complex EM and cleaning validation projects. These are my contact details you can see on the slide now, and as I say, they'll be on the slides when you get a copy. Just a reminder about the remaining webinars we have planned in 2024. For more details of any of these webinars, you can head over to our website where you can register and find out more details about them. There you will also find Tim's monthly regulatory updates, as well as you can access RSSL's team of technical experts with any questions you may have. For those of you who are new to RSSL, who are we? So RSSL are a growing CRO supporting clients with a wide variety of analytical services. We have been around for over 30 years and are based exclusively in the Reading area in the UK, where we have 4 separate sites. For those of you who don't know where Reading is, it's about 40 minutes west of London and Heathrow. Our 4 sites accommodate over 250 scientists working and supporting clients in 12 different technical areas, of which microbiology is just one of them. We support clients globally, and we have clients all around Europe, as well as in the US and as far away as Singapore and Australia. RSSL is a trusted CRO with a big emphasis on quality, science and service, and this can be shown by our high Net Promoter Score. Because our sites sit close to one another in the Reading area, we can show great agility to manage complex multi-discipline projects. We also have dedicated multi-lab project managers and client liaison managers who can help keep those complex projects on track and managed. You may not be aware, but we are backed by a multi-million-dollar company, Mondelez, which allows us to have access to funding to support our expansion and growth as our markets grow and diverge as well. The 12 technical areas that we have specialize in us, allowing us to support specialize in different techniques, allowing us to support a diverse range of biological, microbiological, chemical and physical analytical services across all phases of the clinical development through to commercial release. We are exceedingly well placed to help support you with all of the new Annex 1 regulations and are lucky enough to have a knowledgeable technical team who can advise and support you throughout your manufacturing processes, be that raw material testing, investigative support of any issues you may face, such as impurities or foreign particles. And we have a well-equipped and extensive microscopy department which can help build that particle database for your manufacturing processes. Our microbiological support is extensive and we can support all aspects of your Annex 1 compliance, including microbial identification, today's topic. We have a state-of-the-art MALDI-TOF system which enables us to identify any EM isolates you may have or any unfortunate positives you may find in your bioburden analysis. And we can offer a broad range of turnaround times to ensure that you can get those IDs through in as little as 48 hours sometimes. Well, that's enough of me. I would like to now introduce you to our guest speaker, Dr. Tim Sandle. I'm sure many of you have heard of Tim and subscribed to his many publications. Tim has over 25 years' experience of microbiological research and biopharmaceutical processing. He is a member of several editorial boards and he has written over 600 book chapters, peer-reviewed papers, and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK, and he also finds time to be a visiting tutor at both the University of Manchester and UCL. I'm going to now hand over to Tim and set him up as the presenter.
Okay, thank you very much. I'll just switch over. Okay, is that okay, Annette? Is all good?
Yeah, that's good. Thank you.
Okay, brilliant. Okay, well, thank you, everybody. Thank you. It's a great pleasure to be back with you. And what we are looking at in this webinar is a number of microbial identification related areas. So we're considering why microbial identification is important and what are some of the things we can do with the data that we gather. We'll be looking at how we can develop an identification strategy, answering those questions of what, when, how often and why we're carrying out identifications. Then we'll be having a look at some of the different identification technologies available. Annette's mentioned MALDI-TOF, and that's certainly a key technology, but we'll look at some others as well. We'll also consider questions of outsourcing, particularly if we have a requirement to perform genotypic testing. We'll also consider when things go wrong or flaws with identifications, and a little bit about trending as well. So in terms of characterizing microorganisms, there are a number of situations, scenarios or reasons why we'd want to do this. So this could be to look for changes to the norm. So if we see a particular pattern of organisms consistently and then suddenly there's a shift, this may signal changes to a process, changes to cleaning or disinfection. There might be very serious points of contamination. So if we're a sterile products manufacturer and we have a sterility test failure, then we would need to work out the origin of that, and often we would need to deploy genotypic testing. And if we're a non-sterile manufacturer, then we're going to have particular concerns with particular objectionable microorganisms. Out of limits results, looking for how we might categorize organisms, and also we want to build up a library, build up a microbial library of things that are common and not common, and then be able to draw upon that library, whether we call them environmental isolates, wild types, whatever, in order to have organisms that we can challenge into culture media. We can help to characterize our disinfectants and so on. So there's a plethora of reasons that we can draw upon.
Tim, can I ask you to close the GoToWebinar menu? It's covering up some of the slides people are seeing. There you go.
Oh dear. Okay. I'll pull it down out as far out the way as I can. Yeah. Okay, thank you. So in terms of what identification is, so microbial identification can be defined as the process of microbial characterization through the utilization of a spectrum of pre-chosen tests. And we can then divide that into presumptive identification. So this might be information that we signal from a culture to help guide us for the appropriate identification method to use, but often this is not going to give us a definitive answer. and then we can have more confirmed methods that are going to designate a species level, and then as a minimum we need to be deploying biochemical tests that can help detect the presence or absence of particular sugars or enzymes utilized or byproducts produced by the unknown organism. And to do that, to achieve that level of identification, then we need an approach, a set of methods to do so. So I'm sure many of you are familiar with the concept of staining, but staining can be very useful as a verification tool with some identification methods. It's an important bridging step for allowing us to select the appropriate methods. So with staining, we're creating a wet preparation of a bacterial or fungal colony. We're adding a dye, and some of the information that we will gather relating to size, shape, arrangement will give us that verification, because we can have a problem further down the line. We can misselect something. Sometimes we might only be interested in characterizing certain organisms. So if we're monitoring a water system, we might be of the view that gram-positives are most likely to be from adventitious contamination, whereas the gram-negative organisms may well be more indicative of what is actually in the water. So what we have on the screen, if you're unfamiliar with these, the first slide on the left-hand side is a gram-positive stain. Happens to be a coccoidal shaped organism. Jumping to the right-hand side, we have a gram-negative stain of rod-shaped organisms, and then in the middle we have a bacterial spore stain, and here the vegetative cells are a sort of crimson red, and our spores have been stained with malachite green, and they're appearing green in colour. But beyond basic staining, we have a wide variety of identification methods, and we can split those into three groups. Some prefer two groups, but we'll discuss three groups. So we have phenotypic methods, which are based on morphological, physiological, and/or biochemical features that are expressed by the organism as it grows. We also have chemotaxonomic methods, which are based on the chemical constituents of the cell, like antigen content, cell wall composition, cellular fatty acid. So fatty acid analysis is almost like a separate group of identification methods. And then within this, we can also consider proteomic methods as well, which are based on the whole cell protein content. MALDI-TOF falls in that group. And then we have the sort of gold standard methods, which are the genotypic methods, and these are based on the genetic makeup of the organism, for example, its nucleic acids. So from that, we have identification systems that are available, and principally these are genotypic, which not that many laboratories will have because the technology is expensive, and if you are fortunate to have that, then probably the justification of purchasing that is the level, complexity and sheer number of identifications you're probably conducting. And we also have phenotypic methods and proteomic methods. So we have these sort of different groupings, and within that we have different systems that we can draw upon. And what we're going to do very briefly is just sort of take a journey through those systems. So if we begin with the simplest biochemical type of method available. So on the screen there is an API kit. It was the first commercialized phenotypic method using biochemical reactions that's sort of brought together as one test. So the first API was the API 20E, and that was created in 1969. And the system is still in use today. And essentially, in each of the wells, there's a series of carbon utilization and biochemical reactions. And there are alternatives now. So there's a BB Crystal and also a kit from Remor that does a similar thing. The limitation with these is that it's based on color changes or degrees of turbidity. One of the API kits in particular, the 50 CHB kit, is looking for variations of yellow and orange. So it does require a degree of skill in interpreting it. And any final decision is going to be left to an experienced microbiologist. But there's not a ready way of verifying the data or with capturing the data. So there's quite a degree of subjectivity with the API. The automation of this technology was the sort of next generation of common microbial identification methods. Kind of two or three key examples are the auto microbic system, the Vitex system, and the Omnilog system. And they differ in terms of the way in which they attempt to indicate the likely outcome of an unknown microorganism. So the Vitex utilizes various identification test cards. They're made of microwells containing biochemicals, and separate cards are available for different groups of organisms. So this technology is very reliant upon initial gram stain in order to select the correct card, which is also a prerequisite for the API kit that we just looked at. Whereas the Omnilog system instead uses proprietary redox chemistry, and it's seeking to measure the carbon utilization of microorganisms in a microtiter plate. And that itself is based on cell respiration, and when that occurs, then there's the reduction of a dye that leads to a color change. In this case, it's a purple color change. Now, these types of systems have some prerequisites, so that they do require pure cultures. They require cultures to be young, so ideally within sort of 18 to 24 hours. We have to produce an inoculum of a specific turbidity, and we also have to make sure that we're using the right type of growth medium. So if a type of growth medium is required for the system, or a type of growth medium is recommended for a particular organism, that is quite critical, because each species of microorganism will have a variety of metabolic pathways available to them. And these can be turned on or turned off depending upon the available nutrients and how that organism grows. So it's potential for some organisms not to be sufficiently activated, so we're not getting those growth changes and therefore not getting the phenotypic reactions that we're looking for. Another thing with such methods is that they generally signal the best match. Now, there are certain criteria that can help us with the probability of a successful match, but it is possible to mismatch, and this can happen at a reasonably high rate, and that's partly reflected on the quality and skill of the preparation of the test, and also what the database is that we're actually measuring against. So sometimes if we've got a database that's too clinically orientated, this can lead to misidentifications. As part of that other group of tests, there is fatty acid analysis. An example there is the MIDI-Sherlock system, and this one looks at, in a sense, the sort of fatty acid fingerprint of the organism, and it compares it to various bacterial libraries. And this happens because the arrangement of fatty acids in bacterial cell membranes differ both qualitatively and quantitatively. And this can allow us to differentiate between genre and then within species of the same genus. And we tend to get sort of variants of sort of branch chain fatty acids in gram-positive bacteria, and straight chain fatty acids are sort of more common in gram-negative bacteria. The sort of latest technologies, so the instant fame version of the Sherlock produces a result in sort of around 3 minutes. So it's drawing some comparability with things like MALDI-TOF. And with the automatic phenotypic systems and with the fatty acid analysis systems, a lot of this kind of matching thing that I was mentioning about and how it comes to a suggested organism is this kind of matching is undertaken through a series of diagrams. So there's an example of a diagram on the slide, and this is what's called a dendrogram, and it's looking at degrees of relatedness. The closer the relatedness, the easier it is to perhaps get the wrong organism. The wider, then there's more chance of potentially getting sufficient reactions to point to what we actually want. Okay, so if we move to the MALDI-TOF, and this was the technology that Annette mentioned, and this is a technology that's becoming increasingly common in many laboratories. If they can afford the capital investment, then the cost per test is generally lower and far faster. So what the concept here is that the protein profile of bacteria reflects the genome of the particular strain. Therefore, determination of the cell protein content can play an important role in their identification and subsequent classification. So we can take that principle and then utilize pre-existing technology, and that technology is MALDI-TOF. So MALDI-TOF is an initialism for a fairly sort of longish phrase, which is matrix assisted, laser desorption, ionization, time of flight. And normally then after MALDI-TOF, we also get the initials MS, and MS is for mass spectrometry. And the basis of the technology is to use laser excitation to ionize chemical functional groups that are part of the protein makeup of an organism. These ions are then separated out in a tube, and this tube is called a flight tube. And the lighter the ions that are created, then the faster they travel. And there is a detector that measures the time of flight for each of these ions. And as that data is recorded, it creates a spectral profile for the organism as a mass spectrum, hence the mass spectroscopy. And this is based on differences in the mass charge ratio and signal intensity. So you then end up with this protein profile that's ideally unique to a particular organism, and then we can compare that profile to other organisms in a database, and hopefully we get a match. And this technology is capable of identifying organisms from a pure culture within a matter of minutes. So it significantly reduces the time to result. So we go back to those automated biochemical systems. We were looking at 24 to 48 hours. Go back to the API strip, again, could be 72 hours before we've got a result. We've reduced that down to very, very short time periods compared with other conventional biochemical methods. And generally, if you look at published qualifications of MALDI-TOF, then the method generally stands out as highly reproducible. However, there are some quirks that need to be observed. So the quality of the setup is important, particularly in ensuring that bacterial cultures are not smeared together. It's also important that with the maintenance of the instrument that the plates are cleaned properly between each use. Any markings or residuals that are going to cause havoc with our intended outcome. It's also possible for errors to occur in the variation of the solvent and matrix composition. So we can get errors with our reagents, to issues with the cultural conditions, issues with the organism's biological variation, and sort of other poorly developed quality control strategies. The other set of identification methods that are important are the genotypic methods, and here we're seeking a far more definitive answer as to our organism, potentially down to strain level. And one of these techniques is a PCR-based method. So this utilizes quite small quantities of a microbial sample, and as with other forms of PCR, it seeks to produce a higher yield of the targeted DNA material. So the first step is typically the what's called the thermal denaturation of double-stranded DNA. So this separates two complementary strands and then prepares them for the annealing process. And at this point in the annealing process, primers are attached to DNA strands. The third step is then the primer extension step, where an enzyme is added, and this catalyzes the synthesis of new strands of DNA by adding nucleotides that are complementary to those in the unpaired DNA strand, and you repeat this 20 to 40 times, and when you get to around 30 times, ideally, then you would have increased your single copy of DNA to around 1 million copies. And this large quantity of DNA can then be used in proprietary genotypic identification systems like riboprinters and in or the microseq system. So considering riboprinting, so if we put a bacterial sample through a device called a riboprinter, this generates a riboprint pattern. And again, it's aligning this pattern, this genetic fingerprint that is going to be unique to the organism that we have processed against what is available in a library, or if it's not existed before, then we have something to add to a library to enable future testing. And one of the powers of ribotyping is it's not affected by the bacterial phenotype, because we're targeting a highly conserved region of the bacterial genome. And so it means that factors like growth media, other changes that might impact the way an organism grows, behaves, responds are completely unnecessary. So we have far more accurate identification. So the riboprinter identifies the isolate by comparing the riboprint pattern against different libraries. And we use a measure of similarity, and we're looking in this case on a scale of 0 to 1 of a similarity of greater than 0.85 as the identification cutoff. And that means that we have a high number of similarities of the DNA fragments that we've been examining. If we want to target strain level, then if we're getting a match of 0.9 or above, then we can also then start using the technology for those purposes. And also when we get a match of 0.9 or higher, the software will automatically place that sample into a particular ribo group. So it's either against something that's in the library, so it will say we've got this strain of Bacillus subtilis and it's matching our Bacillus subtilis family within our ribo group, or it'd be down as an unknown. And either way, if we find that organism again, then we have the ability to know we've had that organism again. So if we're trying to track something through the facility or match grade A or grade B, we've got a mechanism to do that. The alternative genotypic systems, as I mentioned, is a system like microseq, which is a genotypic system that utilizes PCR, as we've explained, and then DNA sequencing to identify microorganisms. And it's the nucleotide sequence of the RNA gene that serves as a unique genetic identifier for the microorganism. And here, the bacterial identification is again based on parts of the genome that are not subject to change to the environment or any other characteristics. So it's the first 500 bases of the 16S gene in the bacterium. When we're talking about genotypic testing of fungi, then we're looking at something slightly different. So with fungal genotypic identification, this is based on ITS regions. So these are the nuclear, ribosomal, internal, transcribed spacer regions. And in a sense, what we're doing when we're examining these is that we're still producing, if you like, a formal fungal barcode. And that's all based on the average length of this region, this ITS region, and how that varies amongst different lineages of fungi. And so far, around 160,000 species of fungi have been characterized. We sound a lot, but it's actually a very still relatively small number of the entire fungal kingdom. In terms of future directions, then microarrays are a potential future direction in microbial identification. Now, this type of technology combines the potential of simultaneous species and strain identification, and it's relatively versatile, and it is possible to detect and discriminate different bacterial samples on a single slide. And these microarrays make use of the hybridization phenomena existing between complementary single-stranded DNA strands. So each sample sequence hybridizes to the complementary strand of a probe that we would add, and that can help us to confirm the presence of a particular gene. So this type of technology is generally confined to academia. It could become the next thing that makes its way to the laboratory in 10 or 15 years' time. So we've got all these different methods, and the first thing is that these methods cost very, very different amounts of money. Some require capital equipment. Some go down to levels that for everyday use we would not be interested in at all. But we also have a sliding scale of increasing accuracy. Another thing is that there is no single universal method that is capable of identifying all microorganisms that have so far been cultured and entered the world's culture collections or into the totality of microbial libraries. That there's no one method that will capture all of those. Where species might be changing taxonomy, or where very, very detailed analysis is required, then in all of those cases, a combination of methods has to be used, or what's called a polyphasic approach. So again, that's not something you necessarily want to carry out at laboratory level, but it's more to highlight the complexity of identification, and despite sometimes what may appear in marketing literature, that there is no single definitive test method. Okay, so we have this bewildering array of different systems. If we're in the fortunate position that somebody says we can buy a new laboratory identification system, or we wish to outsource to a particular facility, there's a number of things we should work through. So I'll just put some of these down on the slide. So one of them is, what's the purpose of the system? Are we going to use it for lots and lots of routine identifications? We're trying to put through 30 or 40 a day, or is this for occasional investigation use? So that then leads to throughput. And with throughput, we also need to consider the entire workflow, what preparatory steps are required, not just the instrument runtime. But instrument runtime is also important. So we need to consider time to result. We also need to work out cost per test, because if we have high capital output, we might be able to justify the return on investment if we have a much lower cost per test than what we're using currently. We also need to consider the complexity of the test, like what's the skill level required? Do we need graduate scientists to execute or not? Capital cost, of course, and also then validation as well. So, what do we actually need to get this instrumentation online? But perhaps most importantly, we need to understand what can it actually identify, and what it can identify is going to be dependent upon the database. So we need to be satisfied that the database of the instrument gives us adequate coverage in relation to the microorganisms of interest to our laboratory. And if we're in the pharmaceutical space, then we want to be satisfied it can pick up a number of organisms we might find in pharmaceutical products or within a pharmaceutical manufacturing environment. If it's too clinically biased, maybe we don't have that. We also need to consider facility requirements. So if we're introducing a new technology that is based on PCR, then we will probably need a separate laboratory and very strict entry controls in order to avoid the cross-contamination of our samples. So a genotypic PCR method might just be completely impractical. In other cases, if we're used to a small space where we've been pairing APIs, and suddenly we want to go to MALDI-TOF, then MALDI-TOF is a relatively large instrument. So we need to understand that. We also need to understand the utilities required. We may want our system to be able to connect up to a LIMS system. We need to understand training requirements and other factors. So we have all those selection things. Another important consideration that any busy microbiology laboratory needs to contend with is with devising an identification strategy. So a busy laboratory can rarely identify everything. The only thing the regulations are quite clear upon is for sterile manufacturers under EU GMP Annex 1, it's saying identify all grade A and grade B isolates. But when we come on to non-steriles, or if we're steriles on non-steriles and we have grade C and grade D or unclassified areas, how many do we want to identify? And if we have a strategy that is just based around let's just do everything over action, but we don't have many action levels, then we're probably not doing enough identifications in order to gain an understanding of the characterization of the microorganisms, and then we won't know if we're slipping with cleaning practices, transfer, disinfection, staff behaviours, or so on. Also, within the identification strategy, we also need to consider the level of identification. So as I mentioned earlier, in some cases, if we're looking at water systems, we might just be interested in identifying gram-negatives. In some cases, we might be satisfied with the genus. So we've got Staphylococci. Does it matter if it's Staphylococcus aureus or Staphylococcus epidermidis, for example? But there's going to be other times when we will want to know the species, particularly if we start to try and work out origins, and particularly when we want to work out whether there is a pattern. So are we seeing the build-up of a particular organism in grade C that, if we have weaker airlock controls, could be transferred into grade B? And then there might be times when we want genotypic identification. Let's say we have a sterility failure, a media fill failure, we've got organism on operator's gloved hand, got an organism in the product, we might have found something in a transfer hatch. Are all these organisms related? Are they all actually the same organism? Can we go down to that strain level? And we need similar considerations within non-sterile environments, particularly for where we're screening incoming materials, and particularly where we might have requirements to be concerned about particular objectionable organisms. And remember that in the pharmacopoeia, although certain organisms are mentioned, these are only indicator organisms, and it's all other similar organisms of a particular group that we need to be concerned about. But beyond incoming materials, also in non-sterile manufacturing, we're going to have an environmental monitoring program and maybe some intermediate and finished product testing as well. And we have an absence of regulatory requirements. Often when we're introducing, qualifying a new process or an area, we would start off with a greater level of identification, and we then might reduce that as we gain an understanding, and then look at a level that we can at least understand the points of risk. So we're applying things like asset methodology, or if we need to understand variances in seasonal fluctuation, or with different staffing shifts, and all the things that come together to give us an idea about what's the challenge of the area, how are we controlling those organisms that we might be most concerned about, and how do we tell that? So we now quickly look at some common issues with bacterial identification. So we have visual identification. So there are some seasoned microbiologists who could pick up a plate and say, I can tell you exactly what that is. There are a few, but it's a diminishing number. So visual identification is time consuming. It's often only possible to identify the genus, and often guesses are wrong. You might be able to say that crenated looking colony is a bacillus, but it's probably about as far as you can go. And it's not going to stand up super strong in a GMP environment. But equally, writing down the colony description, writing down the degree of pigmentation, sometimes running at a gram stain, is very useful, even if the systems say you do not need to do gram stains, in order to sense check what comes out. Because if your identification system is telling you you've got an organism that's only ever found in marine water trenches off the coast of Australia, or within half a mile of a volcano, or is a particularly rare, like the plague pathogen or something, it's probably complete nonsense. So it probably isn't what it is. And sometimes going back to say, well, of course, yeah, the gram stain's this, or the colony shape is that, it's definitely a misidentification. It avoids problems, and also gives your identification process more credibility. One of the things always to avoid is putting strange organisms in front of regulators, which they then look up and then they start telling you that you don't know what you're doing. So it's also important that each laboratory understands the limitations of the identification system, to know that some systems are more reliable for certain organisms than others, and to know that sometimes we will get strange results. With other things, so with gram stains, just a few pointers there. So for an effective gram stain, we need a reasonably high concentration of bacterial cells. So we need around 10,000 to 100,000 as minimum. We can mess up gram stains. So gram positive bacteria can appear gram negative due to excessive heat fixation through the use of iodine solution that's too old or through over decolorization. Gram negatives can appear as gram positives if the smear is too thick, or if our cultures are too old. And also we can do improper heat fixing and sometimes merge cultures together. APIs, as we've discussed, are highly subjective and dependent upon looking at color changes and have weaknesses in terms of data integrity. Phenotypic systems, as I mentioned, are all based on probability. So if we're required to use a certain card and we use the wrong card, the system will still try and match to something. Or even if it's a universal test kit, it's still going to say, well, the majority of the reactions are this way and this is the closest fit. It doesn't necessarily mean it's the right outcome. And as I said before, it's always important to sense check. You know, okay, your system may have told us this, but is that an organism that, you know, are we really going to find it in our clean room? The other thing about phenotypic systems is, again, as I mentioned, that the organism is, all organisms behave differently in different environments. So the type of culture media, the incubation temperature, the availability of chemical and nutritional factors are going to influence how that identification comes out. We also need to be careful about the purity of the sample and other things like the pipetting skills of the analyst. With genotypic systems, even here, they're not 100% accurate. And the other thing is that to go for sequencing and matching, that's always quite expensive. So we're always going to have, just to recap, this kind of weaknesses of around databases. And again, something else to consider with databases is when we change between systems, the databases might be very different. So we might see what looks like a shift in our microbial profile, but it might not actually be a shift in the microbial profile. It might just simply be the way that the two different databases are aligned. And we're also going to have taxonomic changes. So we're going to get, as molecular technology advances, and it's whether we're keeping up to date with those taxonomic changes to our databases and understanding what they are. So the common Pseudomonas aeruginosa that we use as a common QC strain is now been reclassified as Pseudomonas para-aeruginosa. So slight difference, but something to keep in mind. So things can go wrong, and there's just a couple of cases there on the slide of what can happen. You know, we can end up with recalls. We can also end up with releasing products and then having to pull them back because we've suddenly realized that the identification result is wrong. And you know, things can happen with raw materials, things can happen with over-the-counter systems as well. We also need to develop a strategy for qualifying microbial identification systems as well. And there are different approaches. My advice would be to focus on repeatability and reproducibility. So as a minimum, we want to run a series of known cultures, which we want to run in triplicate per analyst, and then have at least 3 analysts attempt to screen those as well. It can always become challenging when we're looking at environmental isolates against 2 different systems, and we do need to be careful there. And we also want to run a sufficiently large number of organisms so that we can develop our qualification with sufficient accuracy. But just to close, the characterization, the profiling of microorganisms is really important. We can draw a lot of valuable information that can help to support our contamination control strategies and to meet some of those requirements embedded in things like Annex 1. So, are we finding things that are associated with people like staphylococci or micrococci? This might indicate, cross-contamination from people. Are we finding things that are likely to be transferred in from incoming materials? Are we finding things that might be associated with water or from other parts of the human body? So there's a wonderful array of things that we can draw upon. Okay, so that brings my part to an end, and I'll pass you back to Annette.
Okay, we have run out of time for questions, unfortunately. But what I can do is I've noted down the questions we've been asked online, and we'll take them, as I say, we'll take them offline, and I can send out the answers once we've had Tim look at them in the next sort of week or so. So I'd just like to say thank you for attending. Apologies that we started a little bit late, so we couldn't get any questions going. We had some IT issues our end as well. Just to remind you that we have another webinar coming up in October, actually on Halloween, and that's about detecting and eradicating mycoplasma in ATMP cell lines. If you go to our website, you can sign up for this, and also, as I say, check out Tim's blog or any other information that you may need from us, including if you need us to help with your identifications, you can go on there and see what we can do. So I'd just like to take the time to say thank you for attending, for listening, and wish you the best afternoon for the rest of the afternoon. Thank you.
Thank you.